Welcome to the deep dive.
Great to be here.
So today we're looking at some fascinating science. We've got a key paper from Anjuan Tochemi, some news around it.
And it's really quite something, a discovery that sort of turns a fundamental biological idea on its head.
Right. It challenges what we thought we knew about how life's basic molecules like proteins and DNA interact.
Exactly.
Yeah.
Our mission then is to unpack this. It's a bit like, as one source put it, stepping through the looking glass into a molecular mirror world.
Okay, looking glass, mirror world. That immediately makes me think of molecular handedness, like our own hands.
Precisely. Think about your hands, your left and your right. They're mirror images, aren't they?
Yeah, reflections.
But you can't superimpose them. Try putting your left hand flat on your right hand, palm to palm, left glove on your right hand. It just feels wrong. Doesn't fit.
Right. They look the same in the mirror, but they're fundamentally different in 3D space.
That's chirality, or handedness. And it's absolutely vital in biology. The amino acids that build proteins, the sugars in DNA and RNA, they all have this property. They're chiral.
And life on Earth is, well, famously picky about which hand it uses, isn't it?
Incredibly picky. It's called homochirality. Almost universally, life uses left-handed amino acids for proteins. And the sugars in DNA and RNA, they're almost exclusively right-handed.
It's like a standard template across all of life.
Yes, one of biology's really strong biases. And the assumption has always been, if you mess with that, if you try to build a protein with right-handed amino acids or get a normal protein to interact with, say, mirror image DNA.
It just wouldn't work. The lock-in key wouldn't fit. Like Alice in Wonderland trying to use her key in the looking glass house.
Exactly. The shapes are wrong. The interaction fails. That was the dogma.
Okay, so where does this new research come in? What breaks that mold?
Well, the focus is on a specific protein structure. It's ancient, apparently very simple structurally, and is found everywhere, called the Helix-Hairpin-Helix motif. Let's call it HHH.
HHH. Okay, common structure. What does it normally do?
Its day job, usually, is binding to DNA and RNA. It's involved in lots of fundamental processes because of that.
Got it. So a core piece of the machinery. And the big news is?
The big news is that this little HHH motif is, get this, functionally ambidextrous.
Functionally ambidextrous, meaning?
Meaning it can successfully bind to both the normal right-handed DNA double helix and a synthesized mirror image, left-handed version of DNA or RNA.
Whoa. Wait. So going back to the glove analogy,
Yeah.
this protein is like a glove that fits perfectly well on both a right hand and a left hand.
That's a great way to put it. It works in the normal world and the mirror world, interacting with both forms of DNA.
Wow. That seems to violate the whole lock and key principle we just talked about.
It does, and that's why it's so significant. It's the very first time any protein that naturally binds nucleic acids has been shown to do this, to work in the mirror world. It flies in the face of that core assumption about handedness being exclusive.
That's huge. It feels like something you wouldn't even think to test for. How did they even arrive at looking for this?
It apparently started with what one of the researchers, Liam Longo at ELSI in Tokyo, called a crazy idea.
A crazy idea. Love those.
Right. He was looking at the HHH structure, doing some computational work, and just wondered, could this thing maybe bind mirror DNA? It seemed completely counterintuitive.
But he pursued it.
He shared it with collaborators. Norman Matanis at Hebrew University was key here. And they decided, okay, let's actually test this potentially crazy idea.
So how do you even test that?
Well, first, the hard part. You have to chemically synthesize the mirror image of the protein itself, making a protein entirely out of D amino acids instead of the natural L amino acids. That's a big challenge.
Okay, so they make the mirror protein. Then what?
Then they did the experiment in the lab. They took this mirror image HHH protein and tested if it would bind to normal, standard, right-handed, double-stranded DNA.
The kind of DNA that's in, well, us.
Exactly. And remember, the expectation, based on everything we thought we knew, was it shouldn't. A mirror image protein shouldn't interact properly with natural DNA's right-handed twist.
What?
But it did. They measured binding. It actually stuck to the DNA. Longo's crazy idea wasn't so crazy after all.
Okay. Wow. So the mirror protein can physically attach to normal DNA, but is it the same kind of binding? Or is it just sort of, clinging on randomly, not really doing the job properly?
Precisely the critical follow-up question. Is this interaction meaningful? Is it similar to how the natural protein binds the natural DNA?
How do they figure that out?
They used a couple of approaches. They look at kinetics, how quickly the protein binds, and importantly, how quickly it lets go. the dynamics of the interaction.
Okay, the timing.
And they also did mutagenesis studies. They deliberately changed specific amino acids in the protein structure.
Tweak the building blocks.
Right. And then they saw how those specific changes affected binding, both in the natural protein natural DNA case and in the mirror protein natural DNA case.
Comparing the effects of the mutations.
Clever. And what did all that tell them? Were the binding styles similar?
The results from both methods, the kinetics and the mutations, pointed towards, and this is the researcher's term, surprising similarities.
Surprising similarities. So it wasn't just a weak, non-specific sticking. It seems related to the natural function.
Exactly. The way it bound the parts of the protein that seemed important based on the mutations, it looked analogous. Which, honestly, just deepened the puzzle. How could this be?
Yeah, that really ramps up the significance.
Okay.
So experimental evidence suggests the binding modes are related. Did they try to visualize how it works, like at the atomic level?
That's where the computers came back in, molecular simulations. Professor Yakov Livy's group at the Weizmann Institute took the lead on this. They build these incredibly detailed virtual models.
Like running a simulation of the molecules bumping into each other? Pretty much, yeah. Simulating the physics of how the atoms in the protein and the DNA interact over time.
Yeah.
Gives you a dynamic picture.
And what did the simulations reveal? Did they see how it could fit both ways?
They did. The simulation showed something really interesting. Well, the exact contacts, the precise way the atoms lined up, were definitely different between the natural binding and the mirror natural binding. It turned out that very similar regions of the protein were doing the heavy lifting in both cases. The same functional spots on the HH motif were crucial for binding, whether it was interacting with its normal partner or its mirror image partner's world.
Okay. So the core binding site, the essential part of the protein structure, was basically the same for both jobs, even though the targets had opposite-handedness.
That seems to be the key takeaway. It suggests these two abilities, binding natural DNA and binding mirror DNA, aren't just accidents. They seem somehow linked, built into the fundamental design of this ancient HHH motif.
Well, it's genuinely remarkable. And you said this is the first protein that binds our genetic material shown to do this.
The very first example of this kind of functional ambidexterity for a nucleic acid binding protein. Yes.
Which, as you said, raises way more questions than answers.
Absolutely. The authors are very clear about that. The immediate, massive question is just why? Why does this ancient, fundamental, widespread protein structure have this hidden talent? What evolutionary reason could there be?
Did they speculate? Offer any potential reasons?
They threw out a few possibilities. Maybe the HHH motif needs some inherent flexibility because DNA itself isn't always a perfect double helix. It can adopt different shapes. Binding flexibility might be useful.
Okay, that makes sense for its current job.
Or perhaps this ambidextrous quality helps the protein slide or move along the DNA strand more easily. Another functional possibility in today's biology.
Also plausible. But wasn't there a more out there idea mentioned? Something more radical?
There was. Professor Longo mentioned the one he finds personally most intriguing. The idea that maybe, just maybe, this functional ambidexterity was actually selected for way back in the early history of life.
Selected for? How?
Potentially. And this is highly speculative. Potentially because there might have been other forms of life or pre-life chemistry using mirror image molecules back then.
Whoa! Like a competing mirror image biology. Alice's Looking Glass world was real, chemically speaking.
It's an incredibly provocative thought, isn't it?
Mm-hmm.
And again, huge caution here from the authors. This one study is nowhere near proof of ancient mirror life.
Yeah.
Not even close.
Right. Need way more evidence for that.
Way more. But the possibility that this protein's weird ability is a relic of some ancient interaction with a different handedness. It's certainly exciting for thinking about the origins of life.
So where do they go next? What's the plan?
The hunt is on. Now that they've found one ambidextrous nucleic acid binder, the question is, are there others? Researchers are going to start screening other proteins, other motifs.
See if HHH is unique or just the first example we've stumbled upon.
Exactly. If it's not unique, well, that opens up a whole new layer to understanding how proteins and nucleic acids can interact.
Okay, so let's wrap up this deep dive then. Based on the sources you brought, we've got this ancient, super common protein bit, the HHH motif, that does something amazing. It breaks the rules of molecular handedness. It can bind to both normal DNA and its mirror image.
And simulations suggest this isn't just random. The same core parts of the protein are involved in both interactions, hinting at a deep link between these abilities.
It's the first time we've seen this for something that interacts with DNA or RNA, and it just blows open the possibilities.
It really does. It makes you wonder about the evolutionary path that led here.
So the final thought to leave everyone with. What could this incredible flexibility, this ambidexterity, mean? Does it hint at early life's chemical experiments, or just reveal a whole new dimension of how molecules can function?
It definitely feels like the known universe of protein function just got a little bigger and maybe, a little bit more mirrored. Makes you think, doesn't it?